All right, hello everyone, and welcome back to another chapter of human physiology. So, today we are going to be covering the respiratory system, and this seems like a really logical place for us to go next, considering that we just finished up our unit on cardiovascular. And one of the major concepts that we discussed was the idea behind how deoxygenated blood makes its way from the right side of the heart to the lungs in order to be reoxygenated back to the left side of the heart and then out into the systemic circuit to deliver that oxygen and all the nutrients. So during those discussions, we kind of took it for granted that when that deoxygenated blood makes its way from the right ventricle through the pulmonary arteries and into the lungs, we kind of took it for granted that that oxygenation just kind of happens. So what we're going to do in this chapter is we are going to delve deeper into that particular topic and learn a little bit more about how that actually all occurs. So the chapter objectives here are pretty simple. We are basically going to be covering everything here with the exception of this last bullet point here. What we could cover is kind of the embryology and the developmental biology behind how the lungs and all the different tissues of the respiratory system come to be, starting with the zygote, but this is not a developmental biology class, so we are going to skip over that. So first thing for us to kind of really discuss here is the importance of oxygen in human physiology, and this should mostly be review because dating back to our discussions in Chapter 3, we said that the major function of oxygen for all the different cells in our body is that it allows our cells to maximize their energy production. We did kind of some accounting and determined that a cell that has access to a lot of oxygen can make somewhere between 36 and 38 molecules of ATP for every glucose equivalent that it starts with glycolysis, whereas with anaerobic respiration, we do about 2. So we're talking about a 16-fold difference in efficiency of making ATP. So if you're talking about cells and tissues like skeletal and cardiac muscle, and then also the nervous tissue of the brain and spinal cord, really anything less than maximal energy production is just not going to cut it, especially considering the types of physiological functions that those things do. So we've also discussed back in Chapter 18 how one of the major functions of the blood is to deliver key nutrients and all sorts of hormonal regulators to different cells and tissues throughout the body. And obviously oxygen is arguably the most important of those nutrients. So what we're going to be doing in this chapter is we're going to go through two major concepts. Number one, how is oxygen in the environment, meaning oxygen that is profuse in the air that is all around us, how do we capture that oxygen in our bodies? And number two, once that oxygen is captured in our body, how is that oxygen dissolved in the blood and delivered to all the cells and all the different tissues that need it? So to discuss the first one, how do we capture oxygen in our body, we need to talk about 3 different essential steps in the life cycle of oxygen in your body. Number one, dealing with the capture mechanism. This is called ventilation. So this is the actual mechanical process of capturing air that is in the air around us, capturing that air in our lungs. Number 2 is the process of gas exchange. So the idea here is that once you've trapped that air in your lungs, any oxygen that is present there should be able to diffuse across plasma membranes of involved cells into the pulmonary capillary beds where it should dissolve in the blood or get attached to hemoglobin, as we've discussed before. And at that point, we have reoxygenated the blood, and we can then move that now oxygen-rich blood to the left atrium of the heart. And we know by now what happens after that. So to discuss the second point, which is, okay, once we do have the oxygen captured, what do we do with it? This all has to do with oxygen utilization. How do we deliver the oxygen to all the cells and all the tissues that need it? And of course, this all gets back to cellular respiration. So once that oxygen is delivered to all those metabolically active cells, it gets used by the mitochondria and acts as the last electron acceptor in the electron transport chain of cellular respiration. And of course, it's always good to remember carbon dioxide is produced as a byproduct. And hopefully by the end of this chapter, one thing you will very much come to appreciate is how important carbon dioxide is to the autonomic regulation of our breathing mechanisms. You wouldn't think it would be important, but once we're through this chapter, you're really going to appreciate just how important carbon dioxide is. So speaking of carbon dioxide, any carbon dioxide that gets produced in the course of aerobic respiration is eventually going to need to be expelled through the body. So essentially what's happening here, we breathe oxygen in, we breathe carbon dioxide out. That's probably a little bit too simplistic of a view on it, but that's the basic idea. If that has to be your starting point, that's not such a bad place to start. So what we're going to see here with carbon dioxide is that in addition to oxygen, it is also going to be a very important part of the gas exchange process. And what we are eventually going to see is that oxygen and carbon dioxide tend to switch places anytime gas exchange occurs. Meaning that if you're talking about your pulmonary capillaries, understanding we want oxygen coming into the blood, well, carbon dioxide is probably going to move in the opposite direction, meaning it moves from the blood to the alveolar air, which is something that we'll discuss a little bit later. So here's a little bit of a question for us to ponder for the time being, and this is pretty heavily review, something that hopefully we remember from our exploits into these sorts of things earlier in the semester. So what principle that we have discussed previously do you think is going to dictate in which direction these gases diffuse? If you have one of these gases, doesn't really matter which one. If one of them moves from the blood into the air, or from the air into the blood, from the tissues into the blood, or from the blood into the tissues, regardless of which gas or which direction you're talking about, what is the number one thing that is going to determine which direction, whether it's one way or the other, that gas is going to move? So if you said concentration gradients, you are 100% correct. It's the same deal that we've talked about before with glucose transport, we've talked about oxygen and carbon dioxide transport before, not in this sort of detail, but we've talked about it before. Sodium and potassium movement across a neuron plasma membrane to facilitate action potentials. We've talked about this sort of thing a lot throughout the semester. So for oxygen and carbon dioxide, it's really not any different. So in order to determine which direction one of these gases is going to move, you just have to look at the relative difference in concentration across the plasma membrane. So for example, oxygen is much more highly concentrated in the air that is around us than it is in the pulmonary capillaries. So since oxygen is going to want to move from high concentration to low concentration, oxygen is very much going to want to leave the air that we have breathed in and get into the blood. And like we mentioned, it is usually going to be the exact opposite for carbon dioxide. Carbon dioxide will be much more highly concentrated in the blood of the pulmonary capillaries than it is than the air that we breathed in. So that carbon dioxide is going to want to leave the blood and get into that air that we would then breathe out. So if you look at a quick breakdown of the different types of gases that you find in the air that we breathe, so we're talking about environmental air, the air that's all around us, you'll notice that a good majority of it is nitrogen. So almost 80% of the air that is all around us, 80% of the air that we breathe is nitrogen. Nitrogen is pretty inert. It doesn't really have a whole lot of function in human physiology, so it's not really worth mentioning. We breathe it in, we breathe it right back out. Not that big a deal. Oxygen is at 21%, and at first glance, you might not think that that's a very impressive number. You're probably thinking that's not really a whole lot of oxygen. That is a ton of oxygen, and in fact, that is actually, evolutionarily speaking, the perfect amount of oxygen for us to breathe. Any less, and we're not going to be able to make enough energy for ourselves. Any more, and suddenly you're going to have a lot more free radicals popping up inside your cells, more so than your detoxifying enzymes in the peroxisomes and other places are going to have to be able to deal with it. So 21% is actually perfect for us. Argon at 0.93%, so less than 1%, and carbon dioxide is at less than 0.05%. So really there is hardly any carbon dioxide in the air at all. Now, of course, in recent years you have heard about global warming, carbon dioxide emissions, so that number there may surprise you, and even if that number continues to go up, the fact of the matter is there's very little carbon dioxide, physiologically speaking, in the air that we breathe. So again, when carbon dioxide comes in, the concentration gradient that we discussed here should still apply. So let's take a quick anatomical view of the entire respiratory system. So really what we're talking about here, as we're eventually going to find out, these are going to be the components, anatomically speaking, that are going to participate in the physical ventilation process. How do we actually pull air in, and how do we push air out when the time comes? So we can divide the whole respiratory system up into two major components, the conducting zone and the respiratory zone. Now, the respiratory zone, as we're going to find, refers to one very, very exquisitely specific area. So most of what we're going to discuss here is actually the conducting zone. And the conducting zone is going to make up all the different air pathways that air will take on its way from its point of entry, either the nose or the mouth, and eventually into the lungs, and even part of the lungs itself. So the respiratory zone, as I mentioned, is a very specific portion of the lungs in which gas exchange occurs. So gas exchange is not something that can occur in just any old place. As we are going to find out, there are specific factors that have to be applied in order for gas exchange to occur in any sort of meaningful or efficient way. So let's take a quicker look at the conducting zone for the time being. So the conducting zone, like I mentioned, it basically consists of all of the different pathways that air takes from its point of entry eventually into the lungs. So this consists of the nasal cavity, the pharynx and larynx, the trachea, and then a network of bronchi. You can see here how the trachea, your windpipe, more or less, how the windpipe, the trachea, will branch off into a left side and a right side, which makes sense because you've got a left lung and a right lung. So you've got these bronchial networks, so you've got the two primary bronchi, which then will branch off into secondary bronchi, and those secondary bronchi are going to be what delivers that air, what conducts that air into the actual alveolar tissue of the lungs. So it is worth mentioning with these bronchi, especially the secondary bronchi, they will eventually narrow and narrow and narrow, not unlike what we saw with arteries and veins. They narrow down until they simplify, like arteries and arterioles, veins and venules. These secondary bronchi will eventually narrow down into very thin, about 1 millimeter in diameter, structures called bronchioles. And these bronchioles are actually, it's actually appropriate that we mention it, they're very much like blood vessels. They actually contain circular smooth muscle that, just as the case was with arteries and arterioles, is capable of constricting and relaxing in order to regulate the diameter of the hollow tube, meaning, in this case, with constriction or dilation of these bronchioles, we can either restrict airflow or we can more ably permit airflow. And then the respiratory zone, as I mentioned, this is the very specific place past those bronchioles into what we call the alveolar tissue. This is the very specific place where gas exchange occurs. So we're talking about delivery of oxygen into the blood and export of carbon dioxide out of the blood. So conduction of air from the terminal bronchioles, where we have that smooth muscle, will direct air into these little inflatable sacs called alveoli. So these kind of look like bunches of grapes, and to me, it would not be terribly wrong if you went ahead and just thought of these as little inflatable balloons. When you breathe a volume of air in, these alveoli are going to inflate. When you breathe air out, they're going to deflate to a certain degree. So if you're able to, as you breathe air in, inflate multiple alveoli within one, what we'll call an alveolar sac, you have filled up that alveolus with inhaled air, so lots of nitrogen and oxygen, not much carbon dioxide. So these alveoli are heavily infiltrated with your pulmonary capillaries. So the capillaries that bring in deoxygenated blood via the pulmonary artery and exit out as the pulmonary vein, which of course is going to carry oxygen-rich blood. So the fact that these, as we're going to see, and we'll see better pictures of this that better illustrate this later on, the fact that these capillaries are in such intimate contact with the alveolar tissue is going to be what permits a very efficient gas exchange to occur. Essentially, what we'll see is that oxygen and carbon dioxide, because of a very high surface area, will not have to move very far or really very fast across those membranes in order for the gas exchange that we need to occur very well and very efficiently. So what you're looking at here is you're looking at kind of a different view, a different artistic representation of what the whole gas exchange process essentially looks like. So here with this white arrow, you can see the conduction of inhaled air via those terminal bronchioles and how that is going to direct the air into a single alveolus. So it will be, in reality, thousands, if not millions, of alveoli that all are going to inflate and deflate as individual units. But the other important thing here, as I mentioned, is that each alveolus is heavily infiltrated with capillaries, these pulmonary capillaries. You can see a pulmonary arteriole bringing deoxygenated blood from the right ventricle, and then as we make our way through the capillary bed, gas exchange occurs. We bring oxygen from the air we have inhaled into the blood, which is why once we come out through the pulmonary venule and make our way back to the left atrium, this is now oxygen-rich blood. The detail you are not seeing here is the carbon dioxide content of this blood. So what I recommend doing is I should have by now already recorded and uploaded a separate review video detailing these concentration gradients for oxygen and carbon dioxide and how the whole gas exchange process works in both the pulmonary circuit, which is what you're seeing here, and then also the systemic circuit, which again happens between the left ventricle aorta and then as we make our way back through the vena cava to the right atrium. So definitely check that out if you have not already. So if we look at the cellular makeup of the alveoli, there's really only a handful of different types of cells to talk about. The first and arguably most important of which are called type 1 alveolar cells. So if you picture a single alveolus as being kind of like a balloon, so if you picture just a regular balloon, the wall of the balloon is made up of rubber or latex or whatever kind of balloon that you have. So if you switch gears and say, okay, I've got this alveolus here, what is the wall of this alveolus made of? Well, the answer to that is pretty simple. The wall of the alveolus is made up of these type 1 alveolar cells. These are extremely thin, squamous epithelial cells that, again, are making up the wall of the epithelium of the alveolus. So when an alveolus expands due to inflation, the air is going to push against those type 1 alveolar cells, and this causes it to expand. So as we're going to see here in just a minute, the fact that these type 1 alveolar cells are very squished out and very thin, again, they are squamous epithelia. They have a very thin membrane with a high surface area. This is going to be perfectly suited for our need for efficient gas exchange to occur. The other type of cell that we'll see here, you can see them in yellow here, are called type 2 alveolar cells. These are going to be important a little bit later on, so we're going to kind of punt here and kind of kick this can down the road and promise to discuss these later. So we won't get into it too much right now, but essentially all you need to know for the time being is that these cells are going to produce and secrete a fluidy lipid substance called surfactant, which basically is going to lubricate our lung tissue and allow big changes in our lung shape and volume to occur without any sort of abrasion or any sort of difficulty. But before I move on to the next set of slides, one thing I definitely do want you to take note of here, since in this cartoony representation of several different alveoli, since this is presumed to be a cross-section, hopefully you'll notice that for each one of these roughly 5 or 6 alveoli that are clearly in frame here, you'll notice that the spaces in between them are taken up by capillaries. So as I mentioned and as I promised, capillaries are very heavily infiltrating into alveolar tissue. So if you can picture within each one of these alveoli a whole bunch of oxygen, you can hopefully notice that that oxygen does not have to travel very far to get into a neighboring infiltrating capillary. Same goes for carbon dioxide. So the whole point there, the thing to really take away from that, is that the blood supply and where the air is located are very, very, very close together. So nothing is really going to have to move too terribly far in order to get to where it's supposed to be. So let's talk about what is called Fick's law of diffusion. So this is something that we briefly mentioned all the way back in chapter 3. We didn't necessarily put the whole Fick's name to it, so we didn't exactly know what this is called. But basically what we were discussing are the different types of factors that affect how fast a molecule or a solute is going to be able to move from one place to another across a semi-permeable membrane. So hopefully you guys will remember some of this. So one of the major ones that we talked about is what we called the steepness of the concentration gradient. Essentially, how drastic or how big of a difference is there in concentration of a particular solute from one side of the membrane to another. So on the left side here, you can hopefully appreciate there is a huge, huge concentration of whatever this solute happens to be, not so much on the other side. So the idea there is that because there's a very drastic difference, these solutes on the left side will very quickly move over to the other side, and they will start to move slower and slower as we get closer and closer to equilibrium. Well, this is not the purpose of this discussion here, the steepness of the concentration gradient. It still applies, it's still important, but this is not what we're leading into. The permeability of the membrane to the solute that is diffusing. This is another important one that will determine how quickly a solute can move across. So there's kind of two extremes here. So if this is a cell membrane instead of just a semi-permeable membrane, like what is being demonstrated here with this, I guess it's a fish tank, if this was a cell membrane and these solutes were, say, sodium ions, the rate of diffusion would be 0 pretty much unless there is an open sodium channel, right? So we understand because something like sodium or potassium or calcium, because those things are electrically charged, they cannot move across a phospholipid bilayer unless they have some sort of open ion channel. So that would be a case if you don't have an ion channel where the permeability is so restrictive that the rate of diffusion is practically 0. Or on the other extreme, if you're dealing with something like oxygen gas, carbon dioxide gas, a steroid hormone, cholesterol, something like that, something that because it is totally nonpolar and also very small in the case of gases, because that is completely and freely permeable, it should move at a very high exponential rate because the permeability of the membrane does nothing to restrict it. But again, that is not the purpose of this discussion. But since we are talking about gases, you should go ahead and accept that oxygen, carbon dioxide, even nitrogen, even though it's not that important for us here, because these things are small and because they are nonpolar and they are not electrically charged, you can go ahead and assume that they will freely move across plasma membranes. They will move across the membranes of type 1 alveolar cells without any sort of restrictions. They don't need open ion channels. They don't need carrier proteins. They can move across the membrane just fine. The temperature of the solution, not really that important for us. You will, of course, notice, especially during the winter time, that you labor to breathe a little bit when you're breathing really cold air. And part of that has to do with the fact that the oxygen is just going to move a little bit slower because it's coming out of a very cold solution. But this isn't usually going to be too much of an issue for us. Number 4 here is the most important one that we want to make sure we discuss here, and it's probably the one that we actually discussed the least back in chapter 3, the surface area of the membrane across which the solute is diffusing. So the way I always like to think about surface area is it's all about opportunity. If you've got a whole bunch of solute that is trying to move across a membrane, The surface area represents how much opportunity it has to get across. So if you're talking about a very wide surface area, every single point along that surface area represents an opportunity to get across. But if the surface area is very small and very restrictive, there are only a select few opportunities that solute has to get across. So because greater surface area equals greater opportunity and a much faster rate of diffusion, and because we, of course, understand how important it is to get oxygen efficiently into the pulmonary capillaries so that we can get that oxygen-rich blood to the left atrium, we want the surface area of the membrane across which gas exchange occurs to be as high as humanly possible. And as we mentioned, those type 1 alveolar cells have a very high surface area, so we should be in good shape there. All right, so putting things together with some of the stuff that we talked about in cardiovascular, we've talked about this before, so this isn't anything too groundbreaking. The lungs receive their blood supply from the pulmonary arteries, and then after gas exchange occurs, oxygen-rich blood is then delivered to the left side of the heart through the pulmonary veins. So that's nothing real new. So if we talk about nerve supply into the lungs don't really probably strike you as something that is regulated by the nervous system, but just as most anything else that we've talked about, whether it's the heart, the blood vessels, the adrenal medulla, or other things, the lungs are actually under the regulation of the autonomic nervous system. And here, what we're regulating is the smooth muscle of those terminal bronchioles. We had mentioned that we can either restrict or open up airflow by either closing off that smooth muscle and restricting airflow or opening it up. So for blood flow in arteries and arterioles, we call these processes vasoconstriction and vasodilation. If we're talking about the bronchi, we call them bronchoconstriction or bronchodilation. Now, there's one very important difference to take note of here. When we were talking about vasodilation and vasoconstriction, both of those processes were both mediated exclusively by the sympathetic nervous system. Here, with bronchoconstriction and bronchodilation, it's an either-or proposition. Bronchoconstriction, the narrowing and restriction of airflow, is mediated by the parasympathetic division, whereas bronchodilation is mediated by the sympathetic division. So think of it this way. Bronchoconstriction, a parasympathetic activity, so if you are parasympathetic, you're relaxing, maxing and relaxing, you don't need your airflow to be real high. Parasympathetic activity can slow the flow of air in, especially in cases where your need for air and oxygen is not really that high because you're not really doing anything. However, if you do get to really exercising or running from something, whatever is eliciting your fight or flight response, bronchodilation through the sympathetic division is exactly what you want. You want to open up those airways and get oxygen and carbon dioxide more efficiently transported. So here's a nice little review question, parasympathetic versus sympathetic. I'm not going to give away the answers here. This is going to be something that you'll want to refer back to, I believe, chapter 15 to get the answers to. So these are important things to make sure that you're always aware of. So for a parasympathetic pathway that leads into the bronchioles, is that going to come via spinal nerves or cranial nerves? Same question for the sympathetic division, spinal nerves or cranial nerves. So definitely recommend you look into that and refresh yourself on that particular topic. So here's an interesting thing that you see an awful lot in TV shows, and I'm sure it does happen not all that uncommonly in hospital settings. Anaphylaxis. So anytime a very severe allergic reaction occurs, sometimes along with it you'll get what's called anaphylactic shock. Basically what happens, you see three major things happening to your patient. Their blood pressure drops, bronchoconstriction occurs, so their airways narrow, and the heart rate decreases. So here's my question to you for the time being. Those three things, blood pressure dropping, bronchoconstriction, heart rate dropping, what do those three things all have in common with each other in terms of what caused them? So if you decided that these three things are all the result of parasympathetic activity, then you win. What you win, you don't win anything, all right? I don't have anything to give you, and since this is a virtual class, what am I going to do? Put it in the mail, right? Okay, so anyway, so an overactive parasympathetic division seems to be the culprit here. So an overactive parasympathetic division is dropping your heart rate, dropping your blood pressure, and what is worse here, it is narrowing your airways and making it difficult to get air delivered into the alveoli. So here's the question. If the parasympathetic division is indeed too active, how might we go about reversing that? How can we open the airways up, get the heart rate back up, and get the blood pressure back up? Well, hopefully you remember the balance between parasympathetic and sympathetic activity. So if the parasympathetic division is too active, well, let's just get the sympathetic division engaged. So the common treatment for this, I'm sure you're aware if you've ever watched TV shows like ER or House or whatever, people who are at risk for anaphylactic shock carry around these EpiPens, basically these pens that carry a bolus of epinephrine. Again, epinephrine being one of the major hormones that stimulates a sympathetic response. So intramuscular injection of epinephrine will circulate that epinephrine and it will start binding to alpha and beta receptors all over the place. So this will shift the balance back towards a sympathetic status. So it will raise your blood pressure, raise your heart rate, and most importantly here for respiration, it will ease up the smooth muscle in the terminal bronchioles and allow the airways to open up and clear a little bit. Okay, so let's take a little bit of a different track here. So what we want to start talking about is the process of ventilation, the actual mechanical process of drawing air into the lungs and then when the time comes, pushing the air out. One of the most important anatomical pieces here that is going to be critical to how all of this works is the diaphragm. So we've talked about this before. So the diaphragm, which is this kind of dome-shaped piece of skeletal muscle that you see right here, it basically separates the anterior segment of the body, meaning anything ahead of the spinal cavity, which is not visible here because we're looking at a frontal view. The diaphragm splits the anterior segment into two different cavities. So you've got the thoracic cavity up here, which is where kind of our focus has been for a while now. So it contains the heart. So the heart just kind of sits right on top of the diaphragm in a region which is called the mediastinum. And then bound on either side of the heart is going to be your two different lungs, your left lung and your right lung. And notably, the lungs also sit right on top of the diaphragm. And that's going to be something that's very important to us later on. And then the abdominopelvic cavity down here, everything below the layer of the diaphragm. This is going to contain your kidneys, your digestive organs, reproductive organs, etc., etc. So the lungs themselves will stick up here in the thoracic cavity, since the lungs are kind of going to be our major focus here. The lungs, just as the case was with the heart, we didn't really spend a whole lot of time talking about this with the heart, but just as the case was with the heart, the lungs themselves are contained within a pair of serous membranes, which in this case are called the pleura. In the case of the heart, it was called the pericardium. So if you look at the pleura, so if you look at one of your lungs here, you can see each set of lungs is surrounded by a set of skeletal muscles called intercostal muscles. More on that later. But if you actually look at those muscles stripped away and you're just looking at the pure lung tissue here, then you will notice that if we zoom in, we have, as I promised, two layers of serous membrane. So the pleural cavity in between these two membranes is mostly just an empty, kind of fluidy filled substance. So there's not a whole lot here, so really it's just kind of a thin sheet of fluid that doesn't really contain a whole lot of anything. So that is our pleural cavity, the space in between these two membranes, one of which is called the visceral pleura, and the other is called the parietal pleura. Now, the visceral pleura right here, the innermost, is actually attached to the outermost portion of the lung itself. So the visceral pleura attached to the lung itself. The parietal pleura is attached to the wall of the thoracic cavity. So You have one pleurum, which is attached to the thoracic cavity, the other is attached to the lung. And because there is fluid in between them, it keeps those two sets of pleura kind of together. So the visual analogy I always use for this, and it's usually pretty helpful, imagine that you have two sheets of glass, two thin sheets of glass. Imagine that both sets of glass are bone dry. You put the two pieces of glass together. Are you going to have any trouble separating them apart? No, you shouldn't. They're both bone dry. You should be able to kind of pull them apart. What if instead you have your two sheets of glass and you put some water on one and then stick the other right on top of that? Now, can you imagine really being able to pull those two sets of glass apart? No, because the surface tension on the water is going to hold those two pieces of glass together, you're not really going to be able to do much to separate those two pieces of glass from each other. And that is almost exactly what's happening here with your two different pleura. The pleural cavity here, as I mentioned, contains a lubricating fluid, which, because it is holding those two pleura together, anytime one pleurum moves, the other is essentially going to go with it. And this is going to be important when we start talking about volume changes of the thoracic cavity. When you breathe in and breathe out, essentially what you are doing is you are expanding and retracting the volume of the thoracic cavity. So when you expand the thoracic cavity and it moves this way, it's going to pull your lung tissue out like this, and you're going to force your lungs to expand too, so that we can bring air in. So again, the pleura have a couple of different functions here. I kind of spoiled one of them. So the pleura help to lubricate lung tissue so that any volume changes that do occur aren't going to cause any abrasions or tissue damage. Function number 2, which again I already spoiled, the pleura help to anchor the lungs to the wall of the thoracic cavity so that anytime during the ventilation process, when we cause a change in the volume of the thoracic cavity, the lung tissue is going to behave in the same way. So if we change the volume of the thoracic cavity, we're going to change the volume of the lungs as well. So like I said, best way to think about this, think of two pieces of wet glass stuck together. And here in this bird's eye view that you can see right here, you can kind of see the same sort of thing. So you can see the heart here in the mediastinum. You can see the aorta here. You can see that semilunar valve there. You can see the vena cava here. You can see your pulmonary arteries and veins. So obviously the heart we're very familiar with. Here are the lungs right here. And you can definitely tell that these are the lungs because, again, you can see, you can see the pulmonary arteries and pulmonary veins coming in and out. So what we're looking at here as we focus on the pleura is we're looking at this blue membrane and this green membrane right here. The blue membrane is the visceral pleura and the green membrane is the parietal pleura. And hopefully what you can appreciate from this, the pleural space, that pleural cavity that exists between the two, is mostly negligible in terms of volume. The only thing you should, in healthy circumstances, the only thing you should find there is pleural fluid that holds the two pleura together. Okay, so now what we're going to do is we are going to move on to arguably the most important concept that we are going to tackle in this chapter. So if you've been kind of half asleep up until now would definitely be the time to pause the video, go get yourself some coffee, perk up, because this is going to be super duper important. So question, hopefully you remember this from our cardiovascular unit because it was kind of important there too. What determines the direction in which blood flows? Well, hopefully you're on top of things and you remember that blood flows from areas of high pressure to low pressure. So the answer is pressure differences. So next question, what do you think determines the direction in which air flows? Well, guess what? It's the exact same answer. Air is going to flow from areas of high pressure to areas of low pressure. In fact, when we introduced this concept back in chapter 18 or 19, I can't remember exactly which one it was, the example I used was, imagine that you've got a balloon filled with air. So there you go. So what ventilation is all about is it is this mechanical process of pulling air into or out of the lungs so that air is always following its pressure gradient. We always want air to move from high pressure to low pressure. The idea being, if we want to pull air into our lungs, we want to lower the pressure so that air comes in. If we want to force air out, we want to raise the pressure in our lungs to force it out. So in terms of pressure, this is kind of the annoying thing. There are 3 different types of pressure for us to at least be aware of. Some of them are arguably more important than others. So number 1, the atmospheric pressure, also called piece of ATM. So PATM, the atmospheric pressure. This is the air pressure that's all around us. It's sometimes also called the barometric pressure. So this is something that you can get. You can go on weather.com. You can look up what the barometric pressure is in your area. Usually it should be somewhere around 760 millimeters mercury, thereabouts. So 760 is what it should be at sea level. So if you live in, say, New Orleans, their air pressure should be 760 millimeters mercury. Atmospheric pressure is going to go down the higher your elevation goes. So here in Oklahoma, it's usually going to be more like 750, 752 or something like that. Not a big difference, but a little bit lower. So the next one is called the intraalveolar pressure, or sometimes just the alveolar pressure, so PALV. So this is just the air pressure inside the alveoli. These are the 2 pressures that we are going to concern ourselves with the most. So whether you're breathing in or breathing out, air is going to move from the atmosphere to the alveoli or from the alveoli to the atmosphere. So whichever one of those 2 numbers is higher is going to be where air moves from, and which one is lower is going to be where air moves to. The third pressure, not nearly as important for us, is called the intrapleural pressure. This is just the pressure in the pleural cavity. It's almost always going to be less than the intraalveolar pressure, and that's a good thing because it always applies this negative pressure that allows that pulling force to always be in effect to keep our lungs inflated, which is always a good thing. So the way we are going to affect changes in pressure, now keep in mind that whether we're talking about inspiration, so breathing in, expiration, breathing out, we want air either coming from outside in or inside out. So think about it this way. So first question, if we want to inspire, so we want to bring air from the atmosphere, so that would be your atmospheric pressure, to inside the alveoli, that's your PALV, which one of those 2 numbers do we want to be higher if we want to breathe in? So because air moves from high pressure to low pressure, you would want your PATM to be higher. So if PATM, which again is always going to be around 760, it's not really going to change. There's nothing physiologically that we can do to change the atmospheric pressure, so we want the PALV to be the lower of the 2 numbers. If we want to expire, we want the PALV to be higher. So as I mentioned, the issue is that because we have no physiological means of changing the air pressure out here, all we can really do to facilitate breathing in and breathing out is to change the PALV. What we're going to do to raise the PALV for breathing out and to lower the PALV for breathing in is we are going to mechanically, through ventilation, change the volume of our lungs. And what that's going to require us to do is understand something that you probably have not even thought about since you took kind of your introductory chemistry class. And you're probably thinking, oh, Lord, chemistry, save me from that, right? So what we're going to discuss here is not really that bad. So I promise it's not going to be too difficult here. So you will recall from chemistry that when you talked about ideal gases and the ideal gas law, you talked about several different qualities that gases have. Gases have pressure, they have volumes based on the container that contains the gas. Gases have temperatures, and then there's amount of gas, which is number of moles or N. So 4 different things that we can talk about regarding gases. For us here, because temperature's not really going to be too much of an issue, and neither is moles of gas, everything that we can talk about here essentially will boil, pardon the pun, boil down to pressure-volume relationships. And when you're talking about gases, the relationship between pressure and volume is described by what is called Boyle's law. The idea here is that the pressure on a gas and the volume that the gas occupies is inversely proportional. So what that means for us is that if you increase the volume of the gas, the pressure is proportionately going to decrease. So in this picture that you're looking at here, you have gas particles that are contained within a set space, the volume of which is regulated by a piston that can move up and down. As the piston moves up, the volume gets greater and expands, and the gas, which the amount of which has not changed, the pressure is going to decrease. However, if the piston moves down and shrinks the volume, the pressure is going to get greater because the same amount of gas is trapped within a smaller space, and those gas particles are going to start colliding with the sides of the container more frequently. So if you increase the volume of the gas, the pressure goes down. If you decrease the volume of the gas, the pressure will increase proportionately. So when we say proportionately, we mean proportionately. We mean according to arithmetic, right? So if you double the volume of a container, the pressure is going to go down by 50%, by half. If you reduce the volume by 50%, the pressure is going to double. So these things do happen proportionately. So what our end goal here essentially is to change the volume of the thoracic cavity through the contraction of various muscles, including, namely, our thoracic muscles, those muscles that surround the lungs and are attached to our rib cage and the thoracic cavity, and then also the diaphragm below. So essentially, when these muscles contract, they are going to mold the shape of the thoracic cavity to either shrink it down and increase the pressure, or expand it out and decrease the pressure. So you'll recall that because of the pleura that we talked about, any change in the volume of the thoracic cavity is invariably going to cause the same type of change in the volume of the lungs. So a change in the volume of the lungs, as we discussed with our pressure-volume relationships, is going to produce an opposing effect on the intra-alveolar pressure. So for example, if air is going to enter the lungs through inspiration, we of course understand that we want to get the intra-alveolar pressure below that atmospheric pressure. So we want it to be somewhere below 760 millimeters mercury if you're at sea level. So you might be wondering, okay, how much of a difference does there have to be? How far below 760 do we have to get? So really, it only has to be about 3 millimeters mercury. So if atmospheric pressure is 760, for inspiration, you really just want to get it down to 757. Same deals with expiration. If you want to breathe air out, you want to get your intra-alveolar pressure about 763, about 3 millimeters mercury above atmospheric pressure. And like I said, the transpulmonary pressure, that pressure in the pleural cavity, is always a little bit, the intrapleural pressure is always below about 3 millimeters mercury or 6 millimeters mercury below the intraalveolar pressure. I said you need that negative pressure to always keep the lungs inflated. And then the transpulmonary pressure here is just the pressure applied that is going to get air going the way that it's supposed to go. So a little bit of a case study here. So what I've mentioned is that transpulmonary pressure, or that intrapleural pressure, we always want it to be less than the intraalveolar pressure. Like I said, that negative pressure keeps the lungs inflated, and usually what you want is that transpulmonary pressure to always be positive. If it's positive, you're in good shape. But what happens in cases of pneumothorax, or also called collapsed lungs, is that somehow, some way, we mentioned that the pleural cavity is mostly negligible in volume. There really should only be a small amount of fluid there. If some air somehow makes its way into that pleural cavity is going to fill up with air. The two pleura are no longer connected to each other there. So the intrapleural pressure goes up, and now the transpulmonary pressure shrinks down and is no longer a positive number. So what is happening here is this extra pressure in the pleural cavity will now push against the lungs instead of pulling on the lungs, and this is going to cause the lungs to start to deflate by elastic recoil. So what you're looking at here in this chest x-ray, it's a little hard to tell, but this left lung here is actually okay. You can see the left side of the heart here. It's kind of protruding out because of the size of the left ventricle. The heart is right here. This left lung right here is okay. You have to really kind of look close on the right lung here, but if you look at where I've placed these arrowheads, you can actually see the outline of your right lung right here. I'm kind of tracing my mouse along that. So that is not where the border of the lung should be. It should be attached out here to the wall of the thoracic cavity. So everything that you see in between the tip of the arrow and the back of the arrow is basically air that has come to occupy the intrapleural space. The parietal pleura is still connected to the thoracic cavity. The visceral pleura is still connected to the lung, but there is now a huge separation between those two pleura because a great amount of air has come into the pleural cavity. So think about this for a second. What could possibly cause a pneumothorax? What could possibly cause air to suddenly get from inside the lungs to that intrapleural space? So answers that you could possibly come up with: knife wounds, bullet wounds, anything that opens up or tears an opening into the lungs and allows air to escape into that intrapleural space is a perfectly good explanation. So fixing a pneumothorax obviously is going to involve or entail getting that excess air out and reestablishing a positive transpulmonary pressure to get it to pull back on the visceral pleura to get the lung to reinflate. Couple of things for us to discuss before we kind of wrap this first part of the chapter up. Some physical properties of the lungs which are going to be kind of important moving forward. Things that we want the lungs to have in order for these pressure volume things to apply. Number one is high compliance. So we want the lung tissue to be able to stretch under pressure. So this is measured as a change in volume divided by a change in pressure. Basically, how much stretching or how much of a change in a volume do we really need to get a noticeable change in the pressure? So the idea here is that if breathing is so dependent on changing the volume of the lungs, we need that tissue to be able to stretch under the pressure. Number two, high elasticity. So for these two, for both compliance and elasticity, the visual example I like to use is saran wrap. Okay, so saran wrap or cellophane, whatever you want to call it. So when you're using cellophane, if you say want to wrap up a sandwich, you stretch it out and then you stretch it out to the point where it can fit the whole sandwich inside it and then you wrap it up. But here's the deal with saran wrap. Saran wrap is highly compliant. You can stretch it and stretch it and stretch it, but saran wrap is not at all elastic. You can stretch it, but it's not elastic, so it's not going to snap back into its original shape. Once you stretch it, that is it. It is stretched for good. So when we're talking about the lungs, the tissue of the lungs, it is true that we want to stretch it to accommodate those increases in volume for breathing in, but we also want the lung tissue to be able to go back to normal. We don't want one round of ventilation to permanently change the structure of the lungs. So that would be no good. So thankfully for us, the lung tissue is also highly elastic in addition to being highly compliant. Lastly, we want there to be a low surface tension on the fluid that is contained within the lumen of the alveoli. So this will be something that we discuss a little bit later when we start talking about type 2 alveolar cells, but we'll go ahead and introduce it very briefly here. Basically, if you picture the inside of the alveoli where that air is supposed to be contained, it is basically kept moist by a thin layer of alveolar fluid. Excuse me. So this fluid, because it is mostly water, is going to hold a surface tension. That surface tension, if you picture an alveolus like this and there is fluid all around, that surface tension is going to promote the collapsing of the alveolus, the higher the surface tension is. So because we want alveoli to inflate, we want the surface tension on that alveolar fluid, which again is mostly water, we want that surface tension to be very low. So that is the purpose of the surfactant that is secreted by type 2 alveolar cells. What it does is, because it is a lipidy substance, it gets into the water and it reduces the surface tension by a factor of 15. So 15 times less surface tension means that we're going to have a much easier time inflating our alveoli. So one of the last things we're going to want to do in part 1 of this video series on respiration is we want to kind of reinforce some of these pressure-volume relationships that we've talked about. So during inspiration, we've already covered that we want the pressure inside the alveoli to go down so that we can draw air from high pressure to low pressure from the atmosphere into the lungs. So if we are wanting to bring the pressure down, we want the volume of the thoracic cavity to go up. So we want our lungs to expand. What I really love about these pictures that you're seeing here of 2 chest x-rays that are taken one right after the other, on the left side here you can see a person that has not yet inhaled air yet. So you can actually make out the empty void volume of the lungs here. You can kind of, just based on the dark space there, you can appreciate where the lungs are and what the volume of the lungs are. And then look over here on the right side. This is a chest x-ray taken after the person has taken a big gulp of breath in. And in this dotted line outline here, you can actually see about where the lungs were previously, and now you can appreciate all that extra volume we have added on top of that. We have added extra volume below the lungs. That's going to be due to the diaphragm, and then we've added extra volume on the sides, which are mostly going to be due to the contraction of our intercostal muscles that are pulling out like this, if the diaphragm pulls down like this. So the whole idea here is that if we want to change the pressure and volume of our thoracic cavity and therefore our lungs, it boils down to the contraction of thoracic muscles, including the diaphragm and those intercostals. So there is a little bit of a difference here. There's a little bit of subtlety that we have to understand, the difference between the quiet, ordinary breathing that we do when we're not particularly stressed about anything, the type of breathing you do when you're nice and relaxed, your parasympathetic division is nice and engaged, and then there's forced breathing. So when you go for a run and you're huffing and puffing and drawing big gulps of air in and out, we would call that forced breathing. So there are some subtle differences between the two in terms of how ventilation works. Namely, we are getting more and more muscles recruited into the ventilation process during forced breathing. The more muscles we get involved, the greater the volume change we can apply to the thoracic cavity, and therefore the greater pressure difference and the more air we can draw in and out. So regardless, contraction of these different muscles, the diaphragm is one of the big ones, and then the same goes for the external and internal intercostals. And then there are other ones like your oblique muscles, your scalene muscles, a variety of different accessory thoracic muscles that can all be recruited and applied to get bigger and bigger volume and pressure changes to occur. So I like this kind of side view here in which we can see the diaphragm. So you can see the diaphragm just as it's about to contract and pull down to expand the lungs. And then you can also see when it contracts and becomes more dome-shaped to push the lungs up and decrease the volume. So inspiration, expiration. What is most visible here, obviously, is the contraction of the diaphragm, pulling the lungs up and down to regulate the volume like that. But don't forget, you also have your other thoracic muscles that can pull from the sides as well. So I mentioned that the difference between quiet breathing and forced ventilation are a little bit subtle. So if you look at the types of muscles that are involved, your diaphragm and external intercostals are pretty much going to be involved no matter what. That's going to be how we get the biggest and most significant volume changes to occur, regardless of whether it's quiet breathing or forced ventilation. So during normal quiet breathing, whether it's inspiration or expiration, like I mentioned, you're really only going to get a pressure difference between the alveoli and the environment of about 3 millimeters mercury. And that's perfectly sufficient for your normal, quiet, relaxed breathing. You're not really getting a whole lot of air coming in and out, but you're relaxed. You don't really need a whole lot of air coming in and out. But if your sympathetic division is engaged, you've just ran 5 miles, you're running from a snake or Bigfoot or whatever, something's chasing you, whatever it happens to be, your demand for oxygen in your systemic tissues, your brain, your heart, your skeletal muscles is so high that the amount of oxygen you have coming in via quiet breathing is just not going to cut it. So what we want to do here is we want to greatly increase the rate at which oxygen and air is coming into and out of our bodies. Now, the pressure difference you see here with quiet breathing is not going to be well facilitated for getting air quickly coming in and out. So what do? You get the pressure difference to be even greater. So for forced ventilation, not only are we using our diaphragm and our external intercostals, but we're also recruiting all of those accessory muscles as well. We have more muscles working and we're getting bigger and bigger volume changes to occur. Well, surprise, surprise, we get bigger pressure changes to occur as well. As much as 20 millimeters mercury for inspiration or 30 millimeters mercury for expiration. So the bigger the pressure change, the faster air is going to be moving in and out. And that is going to be how we start to satisfy some of those metabolic requirements when we're heavily engaged in sympathetic activity. Basically, you just get more muscles involved and you breathe extra hard and you get more and more air moving in and out. So the example I kind of laid out, we can see here. Assume you have been heavily exercising. You just ran 5 or 10 miles or whatever it is. Maybe you're a super marathon runner, I don't know. So whatever you want to envision, go ahead and envision that. So assume you've been heavily exercising. So as we discussed, you're going to increase your metabolic demand for oxygen in both your cardiac and skeletal muscle for sure. So if we want to continue to supply a sufficient amount of ATP, we need more oxygen. As we said, normal quiet breathing using just your diaphragm and external intercostals isn't really going to cut it. So how can we possibly get more oxygen into the lungs? So again, as the case was with blood flow, if you can create a bigger difference in pressure between 2 points, meaning between the environment and your alveoli, you can get air to flow much more quickly. So if we are to get bigger changes in the volume of the lungs, past what we normally get during quiet breathing, then the pressure difference will be greater and more air will come in and out. So again, these larger pressure differences are created by recruiting extra accessory thoracic muscles that may not have necessarily been active when you were just sitting on the couch, resting and breathing quietly. So we are going to go ahead and cut off this portion of the video series right here. We'll call this video part 1, and then starting with part 2, we will start with spirometry, the actual physiological measurement of lung volumes and breathing capacity, and then we'll start to lead into homeostatic regulation of oxygen and carbon dioxide. We'll start talking about the autonomic nervous system and how it helps to regulate your breathing rate to make sure that your oxygen and carbon dioxide levels are normal. So we'll go ahead and stop it here for now, and I will see you next time. So sayonara.